First, I'd like to thank the organizers for giving me the opportunity to present this work. And the title of my talk is FeatureREDUCE, Inferring Protein DNA Specificities from High-Throughput Binding Data. So this is an exciting time in the field, and that there are a lot of new emerging in vitro high-throughput protein DNA binding assays. And although FeatureREDUCE has been designed to handle many of these new binding assays, today I'm just going to focus on how FeatureREDUCE produces accurate sequence-to-affinity models from PBM binding data. And the last speaker gave a great intro to PBM, so I'm also going to sort of go a little bit more in depth. So first, PBM stands for Protein Binding Microarray, and it's a binding assay that was pioneered in the Bullock lab. And it starts with a De Bruijn sequence, which is basically a small synthetic circular genome that has been specifically designed to include all overlapping temers at least once, as we see here in part A. And this De Bruijn sequence is then cut up into either 35 or 36 base pair contiguous segments. And then these are used as templates to synthesize the strands on the array. And so if we go here, this step, this is after the synthesis of the single-stranded DNA oligos. And so here we have a single-stranded DNA microarray, and there are about 44,000 spots on the array. And each one of these variable length sequences is attached to the array via a universal primer. And then by using the universal primer, then we synthesize the other strand. So at this point here, we now have a double-stranded DNA microarray. And now we're ready to perform the DNA binding experiment, where we basically throw on purified tertiary factor protein of interest. And fused onto the protein is an alpha GSD tag. And then there's an antibody that recognizes this tag, and the antibody is attached to a fluorophore. And then after the binding reaction has reached equilibrium, then a scanner is used to quantitatively assay the amount of DNA, the amount of protein occupancy at each spot by measuring the amount of fluorescence coming off the fluorophore. So the goal now for FeatureREDUCE and other methods is then to take this raw binding data and to generate an accurate protein DNA affinity model. And the first step in achieving this is to generate a positional independence model, which the previous speaker also spoke about. Now, there are actually quite a few positional independence models out there. The previous speaker spoke of one. FeatureREDUCE actually uses a different one. It uses a position-specific affinity matrix. And this positional independence model was developed in the Bussemacher lab. And it assumes independence between the nucleotide positions. And also, it assumes that each position contributes additively to the overall binding free energy. And what this independence model is basically a matrix of marginal relative affinities. So here in the center, this is a sequence logo, like the ones that were shown previously, where in each column in the sequence logo, we're depicting the level of specificity in each column. And then underneath here, this is the corresponding position-specific affinity matrix for this logo, where for each column, we have listed the relative affinity for that nucleotide at that position in the column. And then to calculate what is the overall relative affinity for a binding site, you just calculate the product of the different relative affinities for each nucleotide at each position. So we can see the relative affinities at each position. They range from 0 to 1. And then, of course, then the product, any possible product, also ranges from 0 to 1. And then using this matrix, you can generate the relative affinity for any possible 10 base pair binding site. Okay. So now, on top of this positional independence model, FeatureREDUCE provides a flexible biophysical framework to incorporate higher order sequence features. It does this by utilizing a robust regression framework that resist overfitting PBM data, which can be very noisy. And the types of higher order sequence features that can be incorporated include dinucleotide dependencies, as well as nucleotide insertions and deletions in the binding site, as well as variable length spacers between the half sites. Secondly, for PBM, FeatureREDUCE also generates a positional bias profile, which normalizes for positional occupancy bias along the probes, which I'll talk more about in the next slide. And so finally, the positional independence model, right, as a basis, and these higher order sequence features combined, we generate a new model, a more robust model that we call a feature-specific affinity model, or FSAM. So what I'm going to do now is I'm going to give an example of how FeatureREDUCE generates an affinity model based on actual PBM data. And the data that we're going to use today is from the transcription factor MIBL2, which stands for MIB-related protein 2, which is a member of the MIB family. And the first step, of course, in generating the model is first the positional independence model. But an important component to building an accurate model turns out to be a positional bias profile. And this is a cartoon that I've put here to sort of illustrate what the positional bias profile is and what drives it. So the analogy I like to use is a bead, right, trying to work its way into a carpet. And the beads correspond to these happy faces and this sad face here. These correspond to nibble proteins, and they're the beads, right? And the carpet is the compacted set of double-stranded DNA oligos that are very near to each other on any spot on the slide. And this cartoon is sort of depicting one spot on the slide, and again, there are 44,000 spots. So the analogy is that these protein/beads, in order for them to reach binding sites that are deep into the carpet of compacted double-stranded DNA oligos, there's an energetic cost, okay? And it's difficult for it to reach these binding sites deep into the carpet of compacted oligos. And because of that, there is a reduction in the relative free protein concentration. Okay? And now down here, this is showing this process of how we generate both the positional independence model and the positional bias profile. So to start, first we need a seed. So we generate the seed by performing a robust multivariate regression over all 8mers. We use both strands because this is a DNA binding protein. Then we again use the robust regression, and we generate a positional independence model. So here is the positional independence binding model for MIBL2. And using just the positional bias, just using the positional independence model, we get actually a very good R-squared in explaining the variance in the binding data. We have an R-squared of 0.64. Now we're ready, now that we have this positional independence model, positional independence model, we now can create a positional bias profile. And we see that there's two things happening here. First, there's a strong bias for binding far from the surface of the slide and close to the free end of the probes. You can see that here. And also, there's a considerable amount of overhang binding on the positive strand. This is showing basically the positional bias profile both for the positive strand and for the negative strand. And now that we have a positional bias profile, we now can go back and then we retrain our position independence model. And we keep doing this until convergence. And then after this is converged, our final R-squared, we see a very significant increase in the amount of the percentage of variance that we explained in the binding data. Okay. Now, one of the things that we notice right away is that we also saw that there was a strong correlation between these positional bias profiles and the amount of R squared to measured MITOMI KDs. MITOMI is another in vitro protein DNA binding assay pioneered by the Markle and Quake labs. And they use microfluidics to measure KDs to particular binding sites. And so here, these R squareds are basically comparing to about 350 MITOMI KDs for these three proteins, CBF1 max and 4,4, which are members of the BHLH family. And we can see here that for CBF1 and max, we see, again, like in the previous example, there's a strong, strong preference to bind far from the surface of the slide and right at the boundary where the free end of the probes are. However, for 4,4, we see the opposite. We see actually that there's a slight preference to bind near the surface of the slide, surface of the array. And we think that this is indicative, actually, of a failed PBM experiment. That what's happened here is that the post-wash, there's been a significant reduction in the post-wash sequence-specific binding in this experiment. Because of that, generates a poor signal-to-noise ratio in the data, and then finally that culminates into a less accurate affinity model. And that's why the R squared to the MITOMI KDs is less for 4,4 than it is for CBF1 and max. So we actually think that these positional bias profiles are actually very good tools to use as a metric to assess the quality of the data and the inferred models. So now, on top of the positional independence model, FeatureREDUCE now uses a robust regression framework to incorporate higher order sequence features. Now, I don't have time to go into all the sequence features that FeatureREDUCE can model, but here I'm showing the nearest neighbor dinucleotide dependency, nearest neighbor dinucleotide dependency correction terms. And so we start off with, again, here's the positional independence model, which is a PSAM, and again, it assumes independence between the positions. And then beneath it, we have a sorted list of dinucleotide correction terms, where a red background corresponds to a term that decreases affinity, and a green term is a dinucleotide dependency term that increases binding affinity. And so if we look at the very first correction term, we see that this dependency term, which is if you have a GC at these two positions, right? This correction term, which is on top of what the positional independence model would predict, we see that this term totally kills binding. Okay? So there's a huge correction term, both with the multiplicative relative Ka and with the additive triple delta G. That is a correction term on top of the positional independence model. And if we look at sort of the first five, right, first five dinucleotide dependency terms, we see that this is really the positions, these two positions minus 1 and 1, this is where the positional independence assumption breaks down, right? Because this is where most of the dinucleotide correction terms are located. And so then this is just showing how the relative Kas, this is the multiplicative factor. So if you have TT, for example, these last two positions, then the correction term is a multiplicative factor of basically 1.6 times what the predicted affinity is when just using the positional independence model by itself. And then similarly, the triple delta G, this is the additive correction term in terms of kcals per mole. Now, one thing we needed to assess the performance of the FeatureREDUCE models. And to do that, we used the MITOMI, measured MITOMI KDs. And again, for CBF1 and max, we are, for each one of these plots, comparing what is the predicted affinity using the PBM data compared to what was the actual measured MITOMI KDs for 350 roughly 350 different binding sites. Okay? And here we're comparing to BEML-PBM, which is another competing algorithm. And we see, first, this is their predictions, and these are their mean squared errors to the MITOMI measurements. And in this column here, this is the feature-reduced position independence models. And we see that we get a significant increase in the mean squared errors for both CBF1 and MAX. And now, after that, we add on the dinucleotide dependency terms that I showed in the previous slide. And we see that for CBF1, there is a significant increase in mean squared error. However, for MAX, there is not. So adding the dinucleotide dependency terms, correction terms, on top of the positional independence model for MAX did not see any significant improvement. So this is interesting, and this correlates with other findings showing that there are dependencies between positions for some proteins and not for others. So finally, another way to validate the performance of feature-reduce is to compare it to the DREAM5 data that was published almost 2 years ago now. And this was a competition that was managed by Stolovitsky and Hughes Labs. The data came from the Hughes Lab, and this is the competition was how well can you predict probe intensities from a PBM design that's different from what you trained on. Okay? And the main performance metric was the Pearson between predicted and actual probe intensities. And so here we show that when adding on the dinucleotide correction terms, we see a significant increase in the mean Pearsons across all 66 proteins. Now, recently, Matt Weirach in the Tim Hughes lab recently did this rather massive comparison study where they looked at 26 different algorithms. And the basis of their comparison was the DREAM5 data. And they also did some in vivo analysis where they compared how well the different models generated correlate with in vivo ChIP-seq and ChIP-exo data. And luckily for me, I don't have time to get into specifics, but luckily for me, feature-reduce came out as a top performer. So this is good in that it shows that we're on the right track. It doesn't mean that we know everything or can explain everything when it comes to protein-DNA interactions and specifically binding affinity, but I think it means that we've made some significant improvements. So finally, this slide, its main purpose is to show that getting these specificities right is very important. In Mark Lundquist's a landmark paper in 2007, they showed that for FO4 and CBF1, both are BHLH proteins that bind to the E box. Their binding motifs are very similar, but there are subtle differences between them. And what they showed was that genomes have evolved to capitalize on these small affinity differences. Okay? That when you look at transcription factors within the same TF family, they all, the logos look very similar, okay, especially to the eye, to the human eye. However, small differences can lead to huge differences in the downstream pathways that are being regulated. So you can see, even though their motifs are very similar, you saw their motifs earlier in the talk, actually they end up regulating very different downstream targets, downstream pathways, because there are small differences in the E-box motifs, right, that have evolved in order to, in the promoter regions of downstream targets, in order to take advantage of the small differences in specificity. So finally, I'd just like to acknowledge really everyone on this slide, especially my advisor and mentor, Harman Busemacher, and also funding from RISE and Magnet and NIH. And thank you for your time.
Really outstanding work. I mean, I'm thrilled that you're able to sort of pick out these very subtle differences. I'm very curious about the faux line being inversely correlated. I'm also curious about the design itself of the different microarrays. I'm wondering if, you know, without knowing that these biases exist, if some motifs are just simply not found near the surface, and maybe that's one of the reasons why these PBMs fail. So my question is, quantitatively, how much of a difference does it make to be near the surface or below? I mean, could you actually just not observe binding at all because you happen to not have, you know, any instances of the motif near the sort of exposed part of the array? And you know, or is it just very subtle? Correct word.
That's a good question. You know, I've looked at a lot of these experiments and the data, and we see a lot of funny stuff. First of all, we see that maybe for the same protein, there'll be, you know, five or seven replicates, and for some of them, there'll be, you know, let me go back up here. Oh, this is good. So for some of them, we get, you know, something that looks like these, which is great. This is what we expect, and most of the experiments look like this, and this means I think that the experiment worked well, right? And then for another replicate, it may look like this. Okay, so even for the same protein, there definitely is a quality control problem.
It may be the same protein, but it could be different peptides, right? Because they're only doing the DNA binding domains in some of these experiments. So, I mean, depending on the size of the domain that you're using, whether you're using the whole protein or just...
Yeah. In that example I just told, I meant the exact same amino acid sequences.
Okay.
So, but you're right. These, as you can see, even for the exact same amino acid sequence, what these positional bias profiles look like is very dependent upon, you know, how big the protein is, what its shape is, okay? Because that sort of determines how well can it get into the carpet of these compact DNA oligos.
And I mean, even the actual positioning of the oligos along the array itself could actually make a difference. I mean, the neighboring probes.
Yes. You know, the designers did their best to try and make sure that there wasn't any cross-hybridization. However, there are known PBM artifacts. And I didn't have time to go into that, but we try and model other PBM artifacts than what I've shown here today. Let's see. I think the two biggest PBM artifacts, one is that there is a known G-content artifact where poly-Gs affect the efficiency of the double stranding of double stranding of the oligos when you're making the PBM microarray. And also, GC content also affects the actual level of reflection of the fluorescence off of the microarray back to the scanner. So what people have done, what we did, is in order to model those artifacts, right, we actually include into the model PBM-specific artifact variables to sort of pick up those artifacts so they don't confound the actual affinity model. And that actually was really the key aspect to really hone and get the best predictive accuracy for FeatureREDUCE.
Okay, thank you.